Good morning friends, myself Dr. Ramesh Bahadure, Assistant Professor, Department of Zoology, Sri Vasantaram Naik Mahavidyalaya, Dharni, District Amravati. Today we discuss about the protein synthesis. This topic brings for BSc third year student. So in that protein synthesis, actually this protein synthesis, that is a central dogma of protein synthesis, we see how the proteins make up all living materials. Proteins are composed of 20 amino acids. Different proteins are made by combining these 20 amino acids in different combination. So in the formation of the chain of amino acid, it attachment to the long polypeptide chain and it form the protein molecules. So this protein synthesis, that is a central dogma of protein synthesis. The DNA at transcription into the mRNA and mRNA translated into the proteins. So in that case, that is a central dogma of protein synthesis. The transcription of the protein and it form the messenger RNA, rRNA and tRNA. How this combining these three RNA, it form the ribosome, and ribosome synthesize the protein and via the translated into the proteins with this flowchart. So comparatively we study the ribosome composition. They are prokaryotic in nature of the protein synthesis and eukaryotic nature of the protein synthesis. In case of the prokaryotes, the 70S ribosomes, the small and large subunit of the ribosome, and in case of the eukaryotic cell, the 80S ribosome, and it differentiates into the 30S subunits and 50S subunits. In case of the 40S subunits, and the separation of the eukaryotes and prokaryotic ribosome composition. Along with their how the small RNA in differentiated into the cluster, the 16S RNA having the 20 plus 21 proteins plus 34 proteins in case of the prokaryotes, and in case of the eukaryotes, the 18S RNA and 28S RNA differentiate into the 25 proteins and plus 40 proteins. This comparative the prokaryotic and eukaryotic nature of the protein synthesis and composition of the ribosome. And the proteins are manufactured made by the ribosomes. So in that outlines of the cells and how they get separated in the rough endoplasmic reticulum and attached the black spots indicates in the form of ribosomes. And this is again the outlines of the ribosomes and synthesis of the newly synthesized proteins and attaches into the colored bits indicates the digitalize the image and to attach one by one amino acid to form the long polypeptide chains, and including the messenger RNA, ribosomal RNA and tRNA. The combining of the three RNA collectively synthesize the proteins. The main function of the proteins help fight to the disease, build new body of the tissues. The enzymes used for the digestion and other chemical reaction are the proteins. The enzymes speed up the rate of reaction. The components of all cell membranes. This is again the outline to how it indicates the protein molecules and synthesize in the plasma membrane layer and differentiating the plasma membrane outside and inside the cell. That is the entry level of the proteins. So this protein synthesis based on the transcription and translation. This is again the protein synthesis flowchart, how it indicates the separation from the DNA to RNA and RNA to synthesize the proteins via the transcription and via the translation. This image indicates how they get DNA gets separate in the form of mRNA. The single stranded mRNA just emerge out from the nucleus into the cytoplasm, and when enter into the cytoplasm at the rough endoplasmic reticulum by brings the amino acid tRNA and the codon and anticodon, they just attach on the mRNA, get processed and synthesize the protein. This diagram indicates the whole protein synthesis protocol. By providing this main basic function through the initiation, elongation and termination pattern. So in the initiation, mRNA binds to the small subunit, the small subunit of ribosomal, and how the mRNA get processed on the initiation. In the initiation, having the initiation factor and keep the large and small ribosomal subunit apart may assist initiation factor 1, 2 and 3. This may assist binding of the initiation factor 2 when they get bind and initiation of the protein synthesis gets started. In case of the elongation, elongated the chain of amino acid by synthesizing the such type of the outlines. In case of the elongation in translation, the P sites, A sites and E sites. The P sites indicate by formation the peptidal sites, A sites, that is the acceptor site with the tRNA bringing the amino acid tRNA to brings the anticodonic region towards this mRNA, and anticodons and codon get bind in the acceptor site. When the acceptor site and peptidal sites get completed, then E sites, that is the exit sites, means where the completion of the protein synthesis, and always the amino acid brings with the methionine. In case of the similar process, just one by one attachment of the amino acid and it forms the long polypeptide chain. After the complete synthesis of the protein, then the termination takes place. The newly synthesized protein, when attachment of the amino acids, leucine, alanine, tyrosine, proline, methionine, the number of the amino acid get attached to each other and release where the termination factor is activated. When terminated the all factors, the tRNA, mRNA, and we synthesize the newly protein, and amino acid having the carboxylic terminus and N terminus at the chemical structure. But at this outline only shows the peculiarly the amino acids one by one attach and it form the long polypeptide chain of the amino acids. So in brief, this step 1, the transcription to making the protein. The first step, copying of genetic information from the DNA to RNA called transcription. So why DNA has the genetic code for the protein that needs to be made, but proteins are made by the ribosomes. Ribosomes are outside the nucleus in the cytoplasm. DNA is too large to leave the nucleus double stranded, but RNA can leave the nucleus having the single stranded. The part of the DNA temporarily unzips, means that is the condense. It is used as a template to assemble the complementary nucleotides into the messenger RNA. So in that transcription process, how the arrangement of nitrogenous bases in the double stranded DNA, when they get uncoiled the DNA, this separates the single stranded in the form of mRNA and attachment of the upcoming ribonucleotides. So again one of the complementary DNA strands and they get separate or unwinding. So simply it synthesizes the single stranded RNA, and on the single stranded RNA again synthesize the number of the nitrogenous bases attached and it form they get complementary nucleotides. And the mRNA then goes through the pores of the nucleus with the DNA code and attaches to the ribosome in the cytoplasmic region. So this process means the DNA convert into the mRNA through the transcription, and when the mature RNA released from the nucleus to the cytoplasm, and transportation towards the cytoplasm and translation takes place. So we already seen that the initiation, elongation and termination process. So this amino acid tRNA brings the chain of amino acid, and in that case the formation of the proteins, and the amino acid along with the tRNA get released after the complete formation of a long polypeptide chain in the form of proteins. Step 2, the translation, the making the actual proteins. In this second step, the decoding of the mRNA into the proteins is called a translation. Transfer of RNA carries amino acid one by one amino acid. The total amino acid 20 amino acid brings one by one from the cytoplasm towards the ribosome. So in that ribosomal unit, and how the mRNA get processed with the anticodon and codonic complementary arrangement, and this UAA, which initiation codon, so just brings the amino acid RNA. This amino acid tRNA brings the amino acid towards the ribosomes, and mRNA having the hosts and anticodonic regions and process the tRNA and arrangement of the amino acid. These amino acids come from the food we eat, we consume. Proteins we eat are broken down into the individual amino acids and then simply rearranged into the new proteins according to the needs and direction of the DNA. These are all the sources of the protein-rich diets. A series of 3 adjacent bases in an mRNA molecules code for a specific amino acid called a codon. Each tRNA has the 3 nucleotides that are complementary to the codon in mRNA. Each tRNA codes for a different amino acids. So in that codonic region, the GCA and the amino acids brings the different 20 amino acids towards the anticodonic region, and the codon and anticodon they get complement to each other. The mRNA carrying the capacity towards the DNA instructions, and tRNA carrying amino acids meet in the ribosome. So this is an animated outlines to indicate how the nucleus, and from the nucleolus they releasing the mRNA having the codonic region in the form of messenger mRNA, and via the transfer RNA, amino acid tRNA brings the amino acid and process on the mRNA. The amino acids are joined together to make the formation of the protein. The polypeptide chains or assembly line through this translation direction, and the completing the polypeptide where the upcoming amino acid tRNA brings the amino acid one by one and from the acceptor side where the peptidal sites means the formation of long polypeptide chains, and after the long polypeptide chain formation they exit into the E site. Use one of the codon charts on the next page to find out the amino acid sequence coded for by the following mRNA strand. So this is the mRNA strand having the complementary codons. So these are all amino acids code for the different proteins, and the all amino acids having the nature like the non-polar, aliphatic, polar, uncharged, aromatic, positively charged and negatively charged. So these are the one base UCAG, and total number of the 64 the codon get appeared through this chart, having the 3 are initiating and 1 are the stop codon. The AUG is the initiating codon, the AAC, GAC, UAA, AUG is code for the methionine, the AC asparagine, GAC aspartic acid, and UAA is the stop codon. So this is the outlines of how the four codes and the genetic code it forms the replicates of the 64 total amino acids. So thanks for watching the video. Please share and like. This is all about the little bit information of the protein synthesis. Thank you so much. So don't forget to subscribe our channel SGBAU ZTA. Thank you.